we're going to move on from cell respiration, thank you Osmond, to cell structure. So as you can see on this diagram here, this is a cutout of a cell. So there's all sorts of different gobbledygook that makes up the inside of a cell. We've already talked about a few of them. Can anyone tell me what this big circle is in the center?
That's a nucleus.
The nucleus. And what does it do?
It's like the brain.
Right, control center of the cell.
Holds DNA.
Holds the DNA.
Okay,
What about that?
Mitochondria.
Mitochondria.
Powerhouse.
Right, it's powerhouse. It goes through cell respiration. And what are all these floating in?
Cytoplasm.
Cytoplasm, right.
not.
Can't really see it because I kind of drew lightly. I didn't expect to have a projector. But there's some rods at right angles to each other. What are those? Covered this last week.
Centriole? Centriole?
Almost.
Centriole?
Wait, what'd you say? Not chromosomes.
Centriole?
Not centrioles. They're centrosomes. Remember?
Nope.
So these, they kind of make it like a plus, and they're rods basically. They will basically tear apart the chromosomes during mitosis meiosis. Can everyone in here working on homework just bring it down just a tiny bit? We're trying to record this. Sorry. Okay. Thank you very much. You can talk, just don't be loud. So we're just going to go through most of these in order. Just kind of number them in here, which one's which. I'll leave this up here for you guys to copy down if you want. And Nathan will take my place partway through. So first off, like I said, is the nucleus. So it has a few sections. Obviously, the outside is the nuclear membrane. It's why there's a nucleus. There's a barrier between it and the outside, otherwise everything would just be free floating. Do all living things have a nuclear membrane?
No.
Yes or no?
No.
No. What doesn't?
Prokaryote.
Prokaryotes, right. That's where we get the name from. Prokaryote, meaning not having that nucleus, as opposed to eukaryote. So essentially, that's just bacteria. So all of the really, really tiny, simple bacteria you're going to run into don't have a nucleus. Their DNA is just free floating in rings. But everything else uses a nucleus. And so inside is all the genetic material. What's that called?
DNA.
When it's just floating around. It is DNA, but what do you call it? Chromatin, right. Because it's just free-floating genes and some protein. There's a blob in the middle here. What's that? Nucleolus. And the function of the nucleolus is to make ribosomal RNA. Essentially, it makes most of what will eventually become a ribosome, which then goes to the nuclear pore and will attach to the endoplasmic reticulum, where it turns into a ribosome. Question?
What are exactly ribosomes?
So if you remember from that last week, ribosomes are the bits where it's chunks of RNA and protein, where you get the messenger RNA that comes along from the nucleus, and that's where proteins are translated from the instructions on the mRNA. So it's like a little ball. It's all throughout the cell and attached to different things. The mRNA comes from the nucleus, passes through there where it's translated into a protein. All throughout the outside of this nuclear membrane, there are ribosomes and nucleopores. Why do you think there are ribosomes right next to all the DNA in the nuclear membrane? Anyone tell me? Speak up, I can't really hear you. Right, not transcribe. But why would they be right there?
Fast transcription.
Right, fast translation. So remember, transcription is when you're copying the DNA instructions into the RNA. So translation is when you translate the instructions from one language, the language of the DNA and RNA, into the language of the protein. So the nucleolus takes up about a quarter of the volume of the nucleus. It's made up of proteins and nucleic acids. this also is not found in a eukaryotic cell that is in the bacteria. One thing about the nuclear pores, I'm going to move up to here. So it's kind of a weird drawing. I'll draw it up here. But essentially, they will regulate the flow of larger molecules in and out of the nuclear membrane while letting smaller ones pass through. So here's kind of what it looks like. So the membrane is twofold. There's two membranes. They'll kind of wrap around here, and then you'll have protein here that will form kind of like a basket down below. And so it's about 120 nanometers wide. The opening is about 100. But the only opening for things to flow through is really tiny. That's 9 nanometers wide. And so what that means is that really big things like messenger RNA, if they try to go through, they'll get stuck. And they are regulated through special proteins that sit on the membrane and help it push through. But smaller things, like ions and water, can just pass right through. No problems whatsoever. So there's a few parts of the membrane. There's the internal membrane.
Jack,
that
could you
holds,
speak up?
What?
Could you speak up?
Yeah. People working at home at the back, can you bring it down just a little bit more? Thank you. Hear me now, Rishi? Okay. So there's the inside membrane. Obviously, that holds all the gobbledygook together. And it also has something on it called the nuclear lamina, something I'll cover in just a moment. Essentially, its purpose is to also hold all of it together and hold its shape. Then there's the external membrane, which is continuous with the membrane of the endoplasmic reticulum, something I'll come to later. Specifically the rough endoplasmic reticulum. And then there's the paranuclear space. That is basically, that's continuous with the inside of the endoplasmic reticulum. So the outside also is dotted with ribosomes. Having two different membranes here, one gives a little bit better protection. Two lets it interact with this space in the rough endoplasmic reticulum, but still regulate stuff, and I'll get to what that is later. And additionally, it lets you specialize what's on the inside versus the outside, or inside versus outside. So the nuclear lamina here, it's kind of like a few squiggly lines. It's attached to the nuclear membrane, and it holds all the proteins that are on that membrane, including these pores. So they'll do special functions, and as interesting as that is, it's not something that's going to be related to UIL, so we're not going to worry about that. If you talk to your biology teacher, they can tell you a lot more. It's the same sort of stuff that would be on the outside of a cell membrane anyways. But there are proteins that will control like what substances are released inside the nucleus and what substances pass to the endoplasmic reticulum. And it also binds to chromatin at specific segments on that DNA called the matrix attachment region. So it's not understood fully yet why or how this works. All we know is that there are non-random sites where the DNA will just bind to this nuclear lamina. And that takes some importance, some unknown importance during chromosomal development. So I'm going to move on. Does anyone have any questions about the nucleus or anything about that? Okay. So now we have the rough endoplasmic reticulum. Can everyone see this picture? Yeah. So you have the nucleus here, and there's a pore there, and this opens up, and it's basically a bunch of, it looks like pancakes stacked on each other. or just flat plates, and they're covered all over with ribosomes. So I'm going to cover those ribosomes first briefly, just a quick review from last week. So they're made out of what? Who can tell me what ribosomes are made of? Two things.
Proteins and RNA.
Proteins and RNA. What kind of RNA? rRNA, ribosomal RNA, and that comes from the nucleolus there. So they're made of 3 strands of RNA and some proteins. The ones that are free floating around the cell, they just make proteins that are used elsewhere in the cell, say in different organelles. The ones that are attached here to the rough endoplasmic reticulum, those basically create proteins that are transported all throughout the other parts of the cell, and that will be secreted to the outside of the cell. They bond to rough endoplasmic reticulum at a spot called a translocon. So that's the site where the ribosomes will attach. And they are kind of randomly attaching and detaching most of the time. If they're basically translating something that needs to go into the RAR, then it will attach itself as it's going and just spit out that protein to the inside. And that brings us to the next bit. So with rough endoplasmic reticulum, all this gobbledygook up there, it holds all the ribosomes, which make the proteins. We covered that. But it's also important in transporting those proteins to the Golgi bodies via vesicles, 2 organelles, which we'll cover in just a second, or just a moment. So the way it does that is it'll mark these proteins with a short chain of amino acids, basically the same thing protein's made out of, and they have these special tags stuck on the end. And everything that wants to move that around will look at that and understand where to bring it. So it'll react with and bind with certain molecules, much bigger molecules, that will transport it to where it needs to go. Any questions about endoplasmic reticulum so far? Okay. So now we have vesicles. So those are basically, anytime you see a diagram of a cell, there'll be like tiny bubbles inside. So if we blow that up, it looks something like this. This takes much longer to draw than I do. So what this is made out of are phospholipids. You don't really need to know much about it right now. Nathan will cover that more when he goes over the cell membrane, I believe. But basically, this is made out of the same exact stuff that the nuclear membrane and the endoplasmic reticulum membrane and the cell membrane and all of that wonderful stuff is made out of. So it's a bubble that will hold some stuff that can fuse with that to move things to wherever they need. And it's important that it can fuse because you can have stuff in the endoplasmic reticulum, and it'll just kind of form a little nub and then bubble off. And then you have a vesicle. So since it binds with itself so easily, it just forms these little bubbles, and then it'll go to the cell surface, bind with that, and release stuff, and then you have secretion. Or with amoebas or like macrophages that eat other things, it'll sort of wrap around it and then pinch together, and then that space wrapped around the membrane will be floating around, and that's called a food vesicle. So they are used in metabolism, transport, buoyancy control of some bacteria, enzyme storage, and chemical reaction chambers. Anytime you need to have something separate from the rest of the cell, and anytime you need to move something, these come into play.
Oh, okay. Well, no, that's okay.
Next is the smooth endoplasmic reticulum. So it's has a similar function to the rough end of